Hi, my name is Kate Clark, and I'm the Technical Director at Microbial Insights. I also have with me Dora Taggart. She's our President and CEO. We're very excited to be here today. I want to thank Rebecca for organizing this and for the opportunity to present for y'all. In the presentation today, we're going to show how molecular biological tools, or MBTs, can be helpful to understand each phase in site remediation. To do this, we'll be walking through the full remediation life cycle at a complex chlorinated solvent site. Also this year at Microbial Insights, our theme is Crossroads. So we'll also have a focus on how these tools can be very helpful in directing how to proceed when you reach a crossroads or a decision point at a site. Before we get into the presentation today, I wanted to start by introducing Microbial Insights in case you're not familiar with us. Microbial Insights is a biotechnology laboratory that was established in 1992 as a technology transfer company based on Dr. David White's research at the University of Tennessee. Dr. White was a phenomenal scientist. He was both a medical doctor and a PhD researcher, and he did some really groundbreaking work to help understand the ways we can transfer the tools from the medical industry to environmental remediation so that we can better understand what's happening inside the black box of our subsurface. So our goal at Microbial Insights is to dive into that black box and use these molecular tools to help us harness the processes that Mother Nature already has in place and to optimize them to efficiently degrade contaminants of concern. It's really our passion for environmental health that drives us to keep innovating and to continue Dr. White's legacy. Microbial Insights has been in business for almost 30 years, specializing in these molecular biological tools. And I wanted to provide a brief overview of some of the common tools. The census qPCR and quantarray analyses can help us quantify microbial populations we're interested in, can really help to illuminate the biological potential for degradation at a site. So if you're testing for Dihelicocoides, we quantify it using census qPCR or as part of the larger quantarray chlor suite. We also offer stable isotope probing, where we can document degradation of hydrocarbons and some emerging contaminants. Compound-specific isotope analysis, or CSIA, can help us document chlorinated solvent degradation. And then there are ways where we can test different amendments in situ. We offer in situ microcosms, so rather than doing a bench scale study, we can do something in the field to test the effectiveness of different treatment conditions so we can understand what's happening before we implement a treatment full scale. And at the start, I mentioned that our analyses are helpful when you reach a crossroads and need to decide which path will move you forward to the goals you have for your site. At Microbial Insights, we always advocate for a multiple lines of evidence approach, interpreting the microbial results in light of what you know about the site history, contaminant trends, and geochemistry like redox conditions. Hopefully, all of these lines will converge to point you toward a viable path to remediation. And I just wanted to talk about ExxonMobil. They're an example of a company that finds molecular data to be helpful in all stages of site remediation. They recognize that subsurface microbiology was often underevaluated at remediation sites, and they recently put together a guidance document for their consultants, demonstrating how molecular biological tools can be leveraged throughout a project's life cycle to cost-effectively advance to site closure. In the presentation today, I want to show an example of site management with using molecular biological tools from inception to remedial selection all the way to performance monitoring. The study site has been an industrial facility for more than 50 years. In 1969, a grass fire engulfed the drum storage area, destroying jumps of waste byproduct from the manufacture of 1,2-dichloroethane, also known as ethylene dichloride. This incident is considered a major contributor to historic groundwater contamination at the site. The groundwater here is naturally saline with TDS around 8,000 milligrams per liter, and it's not used for drinking water, irrigation, or watering of livestock. The site poses a number of challenges for remediation. First of all, there was residual DNAPL and high contaminant concentrations in the groundwater. Historically, the parent compounds 1,12-TCA 1,2-DCA were present at concentrations as high as 1,000 milligrams per liter. Tetrachloroethane was present at about 100 to 140 milligrams per liter. And biotic and abiotic degradation of the chlorinated ethanes produced chlorinated ethenes, including TCE, DCE isomers, vinyl chloride, and ethene. And then while at a lower concentration than the ethanes and ethenes, chloroform was also present and at concentrations of up to 30 milligrams per liter historically. So as you're no doubt familiar with, a mixture of contaminants can increase the degree of difficulty for remediation. Converging pathways can make it difficult to determine which processes are occurring. The substrate interactions and preferential electron acceptors can impact dechlorination rates. For example, in binary mixtures of 1,12-TCA and 1,2-DCA, the halogen ammonium preferentially utilize 1,12-TCA with biodegradation of DCA only occurring once TCA concentrations are low. And then finally, another issue would be the potential for specific inhibition. In this case, chloroform is a known and particularly potent inhibitor of reductive dechlorination of chlorinated ethenes, most notably vinyl chloride. With background sulfate concentrations around 400 milligrams per liter, there's a high concentration of a well-documented completing electron acceptor. As I mentioned, the chloroform concentrations were as high as 30 milligrams per liter. And finally, the groundwater, and I also mentioned before that the groundwater is naturally saline. There is also a non-site source from the former chlor-alkali plant. And then while reports on inhibition by light metals differ considerably, moderate inhibition of anaerobic digestion has been reported at calcium concentrations as low as 2,500 milligrams per liter. So inhibition was definitely a possibility here. Then lastly, the site had many physical challenges. It's a large old source area covering about 7.5 acres. The dissolved plume is also large, extending about 2 kilometers off-site, where it's intercepted by a deep, leaky sewer. The aquifer is a heterogeneous fractured basalt, approximately 20 meters thick, underlain by a low permeability clay layer. And so you can see the size of the natural fractures as well as the clay aquitar layer in these cores pictured. Depending on the extent of the fractures, groundwater velocities can range from approximately 5 to as high as 70 meters per year. So now this site map takes a bird's eye view of the site. The source area includes former surface ponds and the former drum storage area that was destroyed in the grass fire. Groundwater flows generally towards the southeast toward a former 450-meter long ASSVE system that was operated to reduce downgradient migration from the source prior to the establishment of the EISB system. Sections were blasted to increase fracture connectivity and enhance permeability. Particularly in the blast fractured sections, the system was quite successful in contaminant recovery and reducing downgradient contaminant fluxes. The site managers performed extensive chemical, geochemical, and microbial analyses for assessment and groundwater monitoring. So they looked at the contaminant concentrations and daughter products. I won't get into it here, but they also performed CSIA on groundwater samples from select locations. And then the geochemical monitoring included volatile fatty acids, manganese, ferrous iron, sulfate, and dissolved gases. In terms of the microbiology analyses, qPCR and then later quantarray, as the number of targets of interest increased, were performed. So briefly, quantarray is a small volume qPCR system allowing simultaneous quantification of a broad spectrum of microorganisms and functional genes of interest. In this case, we used quantarray to quantify halorespiring bacteria, reductive dehalogenases, and known competitors like sulfate reducers. So here is a contour map like those you commonly see for contaminant concentrations, but this one is for concentrations of the helicoides, during the site assessments that took place in 2011. So dihelicoides concentrations were only greater than the 10 to the 4 cells per milliliter recommended threshold for generally effective rates of reductive dechlorination in the areas of the source zone. The same can be said for Dihelibacter and Dihalogenamonas. Substantial populations were only detected in limited areas under sulfate-reducing conditions. Under the more mildly reducing conditions prevailing across most of the source area, halo-respiring bacteria were detected, but at low concentrations. So to sum up the initial site assessment, there were moderate concentrations of Dihalogenamonas and to a lesser degree Dihelibacter. Dichloroelimination of TCA was producing vinyl chloride under the prevailing subsurface conditions. With dihelicoides and vinyl chloride reductase gene copies low across most of the source area, vinyl chloride was going to accumulate. The only exception was limited areas where the volatile fatty acid production from residual polyglycols led to significant sulfate reduction in substantial dihelicoides populations. So after this initial site assessment, the site managers were looking to evaluate different remediation options. One way they did that was through an in situ microcosm study. These units are an intermediate between a bench scale study and a full pilot study. They give us the ability to test different amendments like we would do on a bench scale study, but we're actually doing it in an open system in the field. And this is what the in situ microcosm units that are deployed look like. Inside each unit is a passive diffusion bag to look at the contaminants of concern, a biotrap for the microbiology, and a VOO vial to look at the geochemistry. Also included are different mechanisms to deliver the amendment we're testing. For instance, with liquid amendments, we can use a sponge. And this setup gives us the ability to look at multiple lines of evidence and evaluate different products in the field with a lower cost than we would see with a full pilot study. After microcosm and pilot studies, full-scale biostimulation was implemented. To date, over 280 metric tons of propylene glycol have been injected. The initial injections were performed as 50% propylene glycol solutions. Subsequent injections were 25% and then down to 10% propylene glycol. Injection solutions were buffered to control for pH. For many injections, what became known as biowater was used to help flush and distribute the propylene glycol. Biowater is simply on-site groundwater extracted from a pumping well that had a high dihelicoides concentration, so it was basically a native bioaugmentation. More than 15,000 cubic meters of biowater and flush water have been re-injected. One injection event also included emulsified vegetable oil and zero-valent iron injected in the air sparge wells along the southern edge of the source area. The propylene glycol injections were carried out at select locations within the former surface ponds and former drum store. However, most were carried out on the perimeter along the upgradient edge of the source zone. So, were they successful in stimulating growth of halo-respiring bacteria? Well, let's take a look at downgradient location where the baseline dihelicoides concentration was near the detection limit. Just looking at this graph, walking through the last 2 years, dihelicoides increased by 5 orders of magnitude following injections. There was about 4 orders of magnitude increase in vinyl chloride reductase gene copies, confirming the potential for reductive dechlorination of vinyl chloride to ethene. Halo-respiring bacteria remained abundant over 3 years after the initial injection. Now, expanding to the whole source area, the impact of biostimulation has become readily evident. Initially, dihelicoides concentrations only exceeded the 10 to the 4 threshold in the western edge, but following the propylene glycol injections, dihelicoides concentrations are on the order of 10 to the 5 cells per milliliter over most of the former ponds, former drum storage, and the source control system. Now looking at the contaminant concentrations, we also saw the effectiveness of the injection. There were decreases in 112 TCA concentrations accompanied by substantial but transitory increases in vinyl chloride, and this is shown for well SVE 130. Although concentrations of 1,2-DCA may have initially increased slightly due to some hydrolysis of 1,12-TCA, ultimately 1,2-DCA was biodegraded, producing ethene. I didn't show it here, but monotonic declines were observed for TCE 11-DCE and cis 12-DCE. Ethene accumulated in the groundwater over the course of bioremediation. So at the site characterization stage, Dihelicocoides and other halorespirators were relatively low, except for sulfate-reducing regions at the northwestern and southern edges of the former surface ponds. After propylene glycol injections, populations of halorespiring bacteria increased substantially, resulting in enhancing anaerobic biodegradation of chlorinated compounds at the site. And at this point, the team had a rare opportunity, almost 300 samples spanning more than 2 years, with quantification of halorespiring bacteria, accompanied by comprehensive characterization of contaminant concentrations and a full spectrum of geochemical conditions. A variety of statistical approaches were then used to identify the key operating variables, to develop target concentrations for these parameters, and then finally to identify any factors that may be limiting or hindering biodegradation. So we started with redundancy analysis and found that out of the geochemical parameters routinely measured, the volatile fatty acid concentrations, sulfate concentrations, and redox had the strongest correlations with concentrations of halorespiring bacteria. So in these charts, we're going to take a look at the impact of these variables individually, starting with the volatile fatty acids. Now here I've shown the results of one-way ANOVA with the VFAs as the grouping variable and the log of Dihelicocoides, Dihellobacter, Dihalogenemonas, and VCRA gene copies as the dependent variables to drill a little deeper. So taking a look at the plot, we see when total VFAs are below about 1,000 milligrams per liter, DHC concentrations are typically below that minimum threshold level of 10 to the 4 cells per milliliter. So as a rule of thumb, we really want to be above that 1,000 milligram per liter total VFAs to ensure that halorespiring bacteria are not limited by low electron donor availability. Once the VFA concentrations are greater than 1,000 milligrams per liter, however, the Dihelicocoides concentrations are greater than 10 to the 4 cells per mil, but the halorespiring populations no longer increase significantly with increasing VFAs. So there's no inherent advantage to this at this site to driving the VFA concentrations even higher. However, we also note that Dihelicocoides concentrations remain high throughout that range. So if site managers chose to use heavy dosing to decrease injection frequency, they could do so without adverse impacts to Dihelicocoides, assuming that adequate buffer is used so that the pH remains near neutral. So here are the same, we did the same ANOVA analysis now for sulfate, which is a known competing electron acceptor. We can see here that in general, concentrations of all the obligate halorespiring bacteria were lower with increasing sulfate concentrations. Concentrations of Dihelicocoides were significantly lower at sulfate concentrations greater than 50 milligrams per liter, and the concentrations of Dihellobacter and Dihalogenemonas were not significantly lower until sulfate concentrations were greater than 250 milligrams per liter. The propylene glycol injection had stimulated growth of high concentrations of halorespiring bacteria, including Dihelicocoides over most of the source area. The lone exception was a distinct area under the former surface ponds where the density of Dihelicocoides remained below the 10 to the 4 cells per milliliter threshold, despite sufficient electron donor availability and consumption of competing electron acceptors. Outside of this area, for example, at well SVE 130, you'll recall that we saw biodegradation of TCA and a transient accumulation of vinyl chloride pretty much right after the beginning of the full-scale propylene glycol injections. In the pocket of low dihelicocoides concentrations, however, that wasn't the case. So here I've shown contaminant concentrations at PZ17 on the edge of this apparently inhibited zone. Maybe there was an initial decrease in TCA and DCA concentrations. The vinyl chloride concentrations did increase, but concentrations of the parent compounds overall did not continue to appreciably decrease. The ethene production here was just a flat line. So now I'm going to turn it over to Dora so she can help explain why we think that effect was happening in this area of the site.
All right, thank you, Kate. So this was really interesting for us because we're trying to figure out what's the difference here that's causing that small area of the site to not be biostimulated like the rest of the area was. So when we looked at this, we could see there's sufficient electron donor in the area, and there's consumption of sulfate. So what's the difference? As we look at these area, we see that on average, the total CVOC concentrations, mainly the chlorinated ethanes and the vinyl chloride, were significantly greater here than in the non-inhibited area. Chloroform concentrations were also significantly higher in this area. And because this area was near the outfall of that former chloroalkali plant that Kate was talking about, we know that while the ionic strength as a whole was not substantially higher, we saw that concentrations of calcium and magnesium on average were higher than outside the inhibited area. So let's take a look and try to figure out what's going on to this. So if we look first at the chloroform, because we know chloroform is a strong inhibitor of reductive dechlorination of chlorinated ethenes. So if we examine the impact of chloroform, again using that one-way ANOVA, on a subset of the data where the growth was not limited by electron donor availability or sulfate, you can see that our dihalocoides concentrations on average are substantially greater than 10 to the 4 when the geochemical conditions were favorable and chloroform was less than 4 milligrams per liter. When the chloroform concentrations were greater than 4 milligrams per liter, we see that the dihalocoides populations decreased significantly to around 10 to the 2 to 10 to the 3. So we know that chloroform is particularly a strong inhibitor of reductive dechlorination based on what we've seen in the literature, and particularly from vinyl chloride to ethene that's remediated and mediated by that vinyl chloride reductase gene. So if you look at that vinyl chloride reductase gene, that VCRA, we can see that when concentrations of chloroform were greater than 4 milligrams per liter, the gene copies were only around 10 to the 1, so a big reduction there. So in groundwater samples with favorable conditions where the DHC were significantly lower at total CVOC concentrations greater than 300 milligrams per liter, however, you can see in these samples the chloroform concentration was 4.6 milligrams per liter in the subset with the total CVOC greater concentrations than 300 milligrams per liter. So this suggests that the lower concentrations may have been a result of the chloroform inhibition rather than toxicity from the high total VOCs. So to understand this better, what we did was we looked at the groundwater samples that had favorable conditions and chloroform below inhibitory levels. And what you can see here is that concentrations of dihalocoides and vinyl chloride reductase genes were not significantly lower even when total CVOC concentrations were greater than 300. So in fact, we could have where dihalocoides and vinyl chloride were high in groundwater samples with VFAs greater than 1,000, sulfate less than 50, chloroform less than 4, even when CVOC ranged from 300 all the way up to 620 milligrams per liter. So, seems like chloroform is the problem that we're encountering here. So how do we overcome this issue? Well, the site manager decided to design a groundwater recirculation system. So in short, flush water was pumped from a non-source area well and injected at an upgradient edge of the former surface ponds. Then biowater with high dihalocoides concentrations were extracted from 3 source area pumping wells and re-injected into 2 injection wells just upgradient of the inhibited area. So this system recirculates groundwater at a rate of about 15 liters per minute, and they could periodically amend it with the propylene glycol if we chose to. So we want to know if it worked. If you look at the Staphylococcus contour map, you can see, based on the sampling event post this, that now all the area, including that formerly inhibited area, is above that 10 to the 4 cells per mil mark. So very successful. So let's take a closer look at the monitoring wells in this inhibited area. The green bars show that the chloroform concentrations decreased from around 8 milligrams per liter in this area to about 4, and then down to about 2.5. Staphylococcus and vinyl chloride reductase gene concentrations also increased by 2 to 3 orders of magnitude. So what impact did it have on contaminant concentrations in that former area? Well, here we've returned to that piezometer PZ17 that was located in the formerly inhibited area. And you can see that during just the propylene glycol injection, it wasn't a notable increase in TCA or DCA. But after recirculation, TCA and DCA concentrations decreased substantially. So do we think it was just dilution? They're just pumping things around? Well, I don't think so, because if it was just dilution, we would expect to see decreases in the concentrations of daughter products as well as the parent compounds. And that's not the case here. Vinyl chloride concentrations, if they didn't increase, they also didn't decrease. And we see ethene concentrations at historic high concentrations in this location. So I'd like to conclude by highlighting the overall impact of this enhanced anaerobic bioremediation at the site. After the propylene glycol injections and the targeted recirculation, the estimated mass discharge through this transect 1 decreased by 80%. This level of reduction has now been maintained for many years. So propylene glycol was an effective electron donor at this particular site. And in terms of site management, looking at all the factors together and using statistical analysis, we were able to understand some site operating variables where we want our VFA concentrations at least 1,000 milligrams per liter. We want our sulfate to be less than 50. So that we can get greater concentrations of Staphylococcus and other reductive dechlorinating organisms. We also found through this that we want our chloroform to be less than 4 milligrams per liter in order to have good DHC response. We also found that targeted groundwater circulation system was able to help us eliminate that chloroform inhibition and overcome that issue. And overall, I was really impressed with this case study because I think it was a good example of how site managers can use aggressive and adaptive site management. You know, the air-sparred soil vapor extraction system that they had was an interim control measure for mass discharge from the source zone. It removed more than 20 tons of chlorinated hydrocarbons from the subsurface for a 7-year period. However, a later passive flux meter survey provided evidence that the system bisected the original NAPL and that the mass discharge was passing underneath that air sparge curtain. Therefore, additional measures were going to be needed to decrease the concentrations. So propylene glycol injections were very successful in stimulating the growth of halo-respiring bacteria and promoting reductive dechlorination in the area. So in that limited area where we saw that biostimulation wasn't successful, again, the site managers were able to pivot and put in that circulation system that helped to overcome that problem. So they continue to adapt and do performance monitoring data from this site to date. So one other tool that we didn't get into today, but I think is really beneficial for reductive dechlorination, is something called compound-specific isotope analysis. This is a way that we can look at the carbon isotope enrichment in our compounds of interest, like in our tetrachloroethene or our trichloroethene, to prove that biodegradation is happening. It's a way that we can get faster site resolution. And my favorite thing about CSIA is that it measures both abiotic and biotic degradation mechanisms. And I think we often leave out that abiotic mechanism that can be an important component for us at some sites. And then not in this particular case, but sometimes we have multiple sources that can be contributing to a down gradient location. We want to do some source delineation to understand who could be responsible. CSIA is a great tool for that, or for vapor intrusion problems as well. So just as a quick summary to help you understand CSIA, and we'll be glad to answer questions on this if you have them at the end of the presentation. This is a slide by Dr. Mike Hyman from North Carolina State University. If you've seen me talk, you've probably seen this slide before because I tend to love M&Ms. This is 13 chocolate fractionation. So we know that in the environment, 99% of our carbon that's stable is a 12 carbon molecule. 1% is a 13 carbon molecule. So the 12 carbon bonds are slightly weaker than the 13 carbon bonds. So microorganisms are going to utilize and break the bonds in the 12 carbon a little more readily. So you're going to start to see an increased ratio of our 13 carbon to 12 carbon present in our sample. So if we look at the diagram here, you can see these little beakers, and let's pretend that the blue M&Ms are our 12 carbon, the yellow M&Ms are our 13 carbon. You see the total concentrations of M&Ms are going down over time. So we want to understand, is it dilution, dispersion, what's happening? Well, because we see an increase in the amount or an increasing ratio of 13 to 12 carbon, or yellow to blue in this case, that means that it's biodegradation that's happening in that subsurface. So it's a quick and easy way for us to understand what's happening with our contaminants in the subsurface. So a quick example of that is if you take a source where we measure the isotopic value. So it's called a delta value, and a delta value is simply your ratio of 13 carbon to 12 carbon compared to an international standard. So if we take that delta value, we measure it in our source area, most of the time these values are going to be pretty negative numbers, like negative 27, negative 30. In this case, it's negative 27. And you can see as we move along the plume that our delta value becomes more positive. So we want to see our delta value increase by at least 2 delta, be more positive by at least 2, which definitely happens in that MW6 and MW7 compared to the source area. So that's definitive proof that PCE is biodegrading as we move down that gradient. If we look at daughter product formation, it can also help us to understand if that's biodegrading. So you can see in this one, if we look just south of or just below the source area in that MW3, our cis-DCE ratio is minus 27.5 delta value. And you can see as we move down gradient, that number really doesn't change very much. Again, we want to see plus 2 delta in order to prove that cis-DCE is biodegrading. So in this case, we cannot say that cis-DCE is biodegrading. We would need to look at that and monitor it over multiple events to see that number become more positive in that same location or in down gradient locations in order to indicate that degradation is in fact happening. So I really like CSIA because I think it's a great way that we can dive into data. And you can also look at more isotopes than I showed you here with just carbon. We can look at chlorine, we can look at hydrogen, and you can do some dual isotope analysis to look at enrichment factors and biotic versus abiotic mechanisms, really glean a lot of information utilizing this data in site management. So as Kate mentioned at the beginning, when you come to an intersection, you got to make a decision. Really looking at all the lines of evidence is really critical and important. So we want to measure the chemistry, the geochemistry, and the microbiology. So we're not making guesses. We're not inferring what's happening. We're actually measuring those numbers. And one of my pet projects in my time here at MI is to start to accumulate all of this information into a database that we can use to help us to get more context and information and be able to make more effective decisions. So at Microbial Insights, we've developed a couple of databases that can be useful to you. The first is our qPCR database. So as Kate mentioned, we've been in operation for 30 years. So we have more than 25 years of qPCR data from over 150 unique organisms and gene targets. These have been collected from over 50,000 field samples. We've done all 50 states in the U.S., all the U.S. territories, and more than 42 countries on all 7 continents now. So a lot of qPCR information. So you can start to look and go, wow, what does my concentration mean? I have this particular gene. Is that high, medium, or low compared to sites around the world? So you can start to put some context to that data and make stronger interpretation. Then we also have the CSIA database, where we've gone through literature and combed over 500 literature values, understanding enrichment factors and isotopic values for a lot of different contaminants. So everything's kind of pulled into one central location. And then this software allows you to go in and to plot the data that you receive for CSIA and make some really interesting graphs that you can use to really help understand what's happening or if shifts are occurring at a site. So we talked about a couple tools today just due to the limited time that we have here with you, but pretty much anything on the molecular side that you guys are interested in, we do it or we work with groups that offer this technology. One of the new things that we've started to offer last year is we acquired Pace Energy, their petroleum forensics division, so we now have that capability to be able to differentiate more on the petroleum side. In characterization, we have some tools that we can use for abiotic degradation, next generation sequencing, pretty much anything on the molecular side. Feel free to reach out to us. And we do a lot of work in the environmental remediation market, but we also look at microorganisms for other processes as well. Same microorganisms are involved in so many things on our planet. So for the oil and gas industry, looking at biofilm formation to protect assets, what's causing corrosion, helping to understand that, to understand if a biocide is going to be effective. We do some work in agriculture, in art restoration. We've looked at everything from King Tut's tomb to preserving the artwork on the Silk Road. We do some work in landfills, biofuels, pretty much anything that's related to microbiological processes other than general health assessment. We are involved in those. And any matrix is easy. So the largest majority of our samples that we get are groundwater samples, and you can see on the left-hand side of your screen there, these are these little field filters that we developed to be able to use in the field. You just pass 1 liter to 2 liters of water through that filter using whatever pump is used to collect the samples and send that filter to us. So it saves shipping liters of water. We can also do analysis from soil, from biotraps, from pretty much any matrix, scrapings from pipes. Pretty much anything that you can come up with, we've tried to extract samples from. We've even had moon rocks from NASA over the years. So pretty much anything you can imagine. And we have 7 locations around the world. Kate and I are based in Knoxville, Tennessee, so that's where our main headquarters is. But we have locations in Canada, Germany, Belgium, Italy, Australia, and China. So no matter where in the world, we should be able to get samples. And we have a great team that's here at Microbial Insights. Kate and I are happy to be here with you today, but you may have heard of Sam Rosalina, who's our CSIA lab director, or Casey Brown, the DNA lab director. And then we have Frank Phillips that does some traveling on sales with us, and then a whole group of staff and technicians that are involved in processing these samples on a daily basis. So at this point, Kate and I will be happy to take questions from you. The easiest thing probably is to go into your GoToWebinar panel, and if you can type any questions that you have into that panel, then Kate and I will do our best to answer those for you. Or if you would like to ask something verbally, you can also raise your hand, and we'll be happy to unmute you so you can ask any questions out loud that you would like to. Okay, the question we have, is this approved by the WDR program? I'm not familiar with that program, but we do a lot of work in California, and we have for the past 30 years. Pretty much any area we've been able to work with the regulatory agencies in that area to give them the information that they needed for approval. Because it's a specialty analysis, it's not widely approved like your VOC or geochemical testing. If you have more questions on that, feel free to let us know. Okay. Looks like we don't have any other questions that are being submitted. If you have additional questions that come up after the presentation, I know it's a lot of material, or if you'd like a copy of the slides, feel free to send an email to info@microbe.com. We can send you a PDF of the slides so you can look at it more, or we'd be happy to answer any site-specific or questions that you may have about this. All right, thank you everyone for your day. I hope you have a wonderful afternoon. And let us know if you need anything additional.
Okay, thank you.
Dora,
Bye.
hello.
Yes. Hi.
Hi,
Can you
this
hear
is
me?
Rebecca. Yeah, just real quick. Could you actually just send me a copy, like a PDF of the slides? I usually throw them into a folder so that our office can take a look at them after.
Okay, sure. Yeah, we'll be glad to do that. Kate will send those to you right after.
Okay, thank you so much. And thanks for the presentation. It was great.
All right. Thank you guys. Have a good day.
You too.
All right. Bye-bye.